Heart disease remains the leading cause of death in the United States. For decades, cholesterol levels, particularly LDL, bad cholesterol, have been a primary tool for assessing risk. However, recent evidence strongly indicates that a biomarker called C-reactive protein, CRP, is a more accurate predictor of the risk for heart attacks and strokes than cholesterol alone. In recognition of this, the American College of Cardiology issued new guidelines in September 2025, recommending universal screening for CRP levels, alongside traditional cholesterol measurements. CRP is a protein produced by the liver in response to inflammation in the body, which can be triggered by infections, tissue damage, chronic conditions like autoimmune diseases, or metabolic issues such as obesity and diabetes. A simple blood test can measure it. A level below 1 mg/dL indicates minimal inflammation and lower heart disease risk, while a level above 3 mg/dL signifies elevated inflammation and higher risk. Studies show approximately 52% of Americans have elevated CRP. The significance of CRP stems from the central role inflammation plays in heart disease. Inflammation is involved at every stage of atherosclerosis, the buildup of fatty plaque in arteries. When blood vessels are damaged, for example high blood sugar or smoking, immune cells infiltrate and engulf cholesterol particles, forming plaque. Over decades, this inflamed plaque can rupture, triggering a blood clot that causes a heart attack or stroke. Therefore, cholesterol is only one component. The immune system's inflammatory response is a critical driver of the disease process. While CRP is a powerful predictor, cholesterol assessment remains relevant, but with a refined understanding. The total amount of LDL cholesterol is less important than the number of LDL particles circulating. More particles mean higher risk, even if the total cholesterol mass is the same. A test for apoB B, apoB, measures this particle count and is thus a better risk indicator than standard LDL measurements. Like CRP and cholesterol, apoB levels can be influenced by lifestyle. Another important marker is lipoprotein A, or Lpaa. This genetically inherited protein attaches to cholesterol particles, making them stickier and more likely to contribute to plaque. Lpa is a strong predictor of risk, but unlike other factors is not influenced by diet or exercise and needs only a one-time measurement. Importantly, lifestyle choices significantly impact these modifiable risk factors. Diet, exercise, and weight loss can lower CRP levels. Foods rich in dietary fiber, beans, vegetables, nuts, seeds, berries, olive oil, green tea, and omega-3 fatty acids, chia seeds, flax seeds, are particularly beneficial. These same factors, along with reducing sugar intake, also help decrease the number of harmful apoB particles. In conclusion, preventing heart disease is more complex than merely avoiding dietary cholesterol. A comprehensive approach involves understanding a combination of biomarkers: LDL cholesterol, CRP for inflammation, apoB for particle count, and Lpa for genetic risk. This complete picture can better motivate long-term commitment to the fundamentals of prevention: a nutritious diet, regular exercise, adequate sleep, stress management, maintaining a healthy weight, and avoiding smoking.